This is TWIS. This Week in Science, episode number 961, recorded on Wednesday, February 21st, 2024. This is possible thanks to a virus. Hey everyone, I'm Dr. Kiki, and tonight on This Week in Science, we will fill your heads with ADHD, bowls, and propagandist AI. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer. The following program is for anyone interested in the world around them. The history, the future, the inner workings of complicated things. This is a place for the curious, the clever, and the ever-restless mind. A sanctuary of reason, a walled garden of delightful thoughts, an ivory tower of intellectually intrepid thinking. From here, you can see it all. Above the clouds, below the ground, everything in between, everything beyond, eliminating the unreal to reveal the real, the actual, the true, the verifiable, the ultimate mirror of existence reflected off the unbiased surface of scientific method. It is time once again to talk about everything that is going on this week in science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki.
And a good science to you too, Justin, and everyone out there. Welcome to another episode of This Week in Science. We are back again to talk about things, stuff, whatevs. Oh, Justin's leaving. Science! We're here to talk about science, as we are every week. Oh, if you are curious about the world, if you want to find a place where we're not just going to give you the pat answers and just whatever it is, we're going to make you think. We're going to give you a little uncertainty. We're going to give you questions. That's what this show is about. We want you to get those brain muscles exercising, because they exercise. Not enough in the world. But I know this audience has big brain muscles. I have stories this week about Saturn. Well, a planet and not a planet. I've got also bowls and balls, propagandist AI, and some brain news. What do you have, Justin?
Oh, what have I got? I've got ADHD. And have had it perhaps since the beginning of humanity. Let's see. There is... a question. Is it even okay to be publishing Chinese research? A Danish bog body has begun to tell its own story.
Dead men tell no tales.
Except for this one. This one's really blabbering on and on. And... some German linguistics professors looked at the numerous ways in which Americans communicate getting drunk.
Communicate getting drunk?
Uh-huh.
Like the words used for, or I can't wait to find out what you mean by communication here.
Yeah.
How do I let you know? Communicate... Let's get smashed, hammered, whatever.
That's like this story right there.
Okay.
It's totally get gazeboed.
What?
Basically, I'll just do the story now.
No,
basically,
No, you teased it. We're going to basically get to it when you get to it, because that'll be fun. First, I need to tell everybody that this is a weekly podcast. We broadcast live on Facebook, Twitch and YouTube, 8 p.m. Pacific time-ish. And you can subscribe to us there, get notifications when we go live. You can also find us as a podcast. Most places podcasts are found. Again, This Week in Science. We're also known as TWIS. And if this is just a lot of stuff to remember, go to our website, TWIS.org, and you can find our show notes, links to stories, all sorts of other stuffs, links to the podcast episodes. But it's time for the science right now. Yeah? Let me introduce the first story to the show tonight. Life and Saturn. Well, Saturn itself, the planet,
what?
we don't really think of Saturn, a gas giant with rings, as a place that is hospitable to life. But many researchers through the years have been like, the moons of these gas giants, perhaps, as they're covered in ice and contain these liquidy mantle center, I don't know, ice encrusted oceans of whatever. Maybe there's life under the ice. And we crashed the Huygens probe many years ago with the Cassini mission to Saturn into Titan, the largest moon of Saturn. And we've also taken many looks at it, and we're like, oh my gosh, Titan, it's got a subsurface ocean, and look at the surface, it's got like mountainy things and valleys, and it looks like it's great, and it's got all sorts of carbon, organic molecules on the surface, amazing. And so from there, everybody just speculated, like, well, if there's so much organic material on the surface of Saturn, then not Saturn, on the surface of Titan, well, then of course, there must be a whole bunch of organic material under that crust and within its interior oceans. Well, a new study out this last week has determined that it's very unlikely, actually, that there's much life underneath the crust. And
Wait. Now, when you say it's unlikely that there's much life, that means there could be a little life.
It could be a little bit. Okay, so they went, the surface is covered in organic material. And hey, these meteors come around occasionally and comets crash into the surface, and they crash in the surface, and wherever they hit, it heats up, and then there's little melty stuffs. And wouldn't that melty stuff have sunk down and gotten incorporated into that subsurface ocean? And anyway, they decided that based on the number of impacts that could have happened, the amount of organic material that's on the surface, and the melt rate melting down into the ocean rate, they decided that the quote is, there's about 1 elephant's worth of organic material in the entire volume of that ocean under Titan's surface. So
you heard it here first, folks.
Yeah.
There's an elephant on Titan.
It's not an elephant.
That's what I heard.
Well, it's approximately the same mass as a male African elephant.
And if you find
glycine.
one elephant, there's probably 2 or 3 more living in the walls.
Yeah. One of the most basic amino acids, glycine. There's no more than about 1 male African elephant in that giant subsurface ocean, which is much, much larger than the volume of Earth's ocean overall. And so 1 elephant in an ocean is not enough to sustain life, according to the lead author of this paper published in Astrobiology, Catherine Neisch out of Western University.
But if the idea, though, is that this organic material got maybe deposited there?
Yes.
Then where did it come from? Is there just organic material floating around on comets and asteroids floating around in our solar system?
Yes. And so the issue is the transfer of the carbon from those asteroids and comets to the surface of these icy moons and then into their subsurface oceans. So how do you make it all happen in the right way? The team and their research, they suggest that it is very likely that none of the icy moons around Jupiter or Saturn, Uranus, Neptune could potentially host life. That this possibility of reduced movement of organic matter from the surface into their oceans isn't sufficient and isn't going to work. So the question is Titan unique in the fact that it's covered in organic material, or is it just the outlier in that other icy moons are more likely to have incorporated the carbon-based molecules into their oceans? I don't know. But anyway, they're going to go look at it. They have a Dragonfly mission, and this researcher, Dr. Naish, is involved in the Dragonfly mission from NASA, which is going to be going out about 2028. And it'll be studying prebiotic chemistry on the on Titan. They'll see. They'll go. They'll sample. They'll see what's happening and let us know what's going on.
Stay tuned. We'll have an update in 4 years. And
In 4
5,
years.
6. How long does it take to get there once they launch?
It was like, what was it, like 13 years?
Oh, gosh.
Something like, I
It's
don't
going to
know.
be a while.
It's a long time. Anyway, it'll be a little bit of travel time. The other Saturn I wanted to talk about is a process. It's a deep learning process that researchers publishing in Nature Methods this last week. They did preprints last year. The Saturn stands for Species Alignment Through Unification of RNA and Proteins. It's a deep learning approach, and what they're doing is taking RNA sequencing, so taking these gene expression of RNA and also protein sequences of known proteins, correlating them with given weights to like what the proteins actually do and their functions, and creating a database that puts together everything so that we finally will not just have like, oh, look, we've got genomes for fish, genomes for birds, genomes for all these different species, but actually starting to put the gene to the protein to the function and be able to get that homologous cell function across species. And so in doing the work that they've published, they've shown that there are a lot of protein types that are related but have moved on, or gene types that are related that have moved on to different cell types and different places in different organisms throughout evolution. On the other hand, they've also shown that there are lots of proteins that might kind of be different in different species, but they have these similar functions. And so they're grouped together based on their function. And I know we've talked about hormones and other active molecules that are named differently in different species because they've been studied for different things, that then they turn out to have the same function, but they're slightly different in their form. But now this is an effort to kind of put all of it together into an atlas using RNA, protein sequencing, and protein function. And they've come up with what they call macrogenes, which is basically like genes that are all kind of might be found in different places, but they all kind of are in charge of the same kind of things. But the whole, the emphasis of this is to see really how much change has happened throughout evolution, from species to species to species.
Very cool. This is pretty exciting, because this could be another way of finding interrelatedness if we've somehow missed it in the genome. Finding
Exactly.
proteins that are doing similar functions might give a hint, although it should kind of be there in the DNA. Also, there's also some. I think we already talked about it on the show, but AI that is starting with function and working backwards to create the protein. We've for a long time been sort of
Right. It's a
generating
good,
proteins and then trying to discover what functions they might have
yeah.
for unique and novel ones. But AI is now at the point where it's like, you want me to design one? Tell me what you want it to do
I'll
first.
make you a protein.
I'll reverse engineer the whole thing. So for at that point, we
Yeah.
should be able to pretty quickly, I would assume, find the function of proteins across the animal kingdom. We should be able to see what it interacts with without having to step into the laboratory.
Yes. Not take things apart. And like right in line with all of this, there's also a group of papers that have been published in Nature Communications Biology and Nature Medicine about what's called the All of Us program. It's run by the U.S. National Institute of Health in Bethesda, Maryland, and it is an effort to create a genome database and is actually trying to improve the way that genomes and health genome data is used to create better health profiles and treatment for all individuals, and is a huge effort to start to represent underrepresented groups. And in this group of papers that have been published, they have published analyses of up to 245,000 genomes that have been gathered by this All of Us program. And in it, there are about 275 million new genetic markers that hadn't been seen, and about 150 may be have some kind of a role in type 2 diabetes. There are gaps that are being filled, and it's pretty exciting. The program has received about $3.1 billion to date, and has been working since 2018 to get people to enroll and allow their genomes to be sequenced. And they had about 100,000 genomes at first, and they've enrolled about 413,000 anonymized participants, and 46% are minority, racial, or ethnic group. UK Biobank, 88% are from white people. So this is a huge repository giving more information about people from different backgrounds, and it can really change the way that we look at various SNPs, or single nucleotide polymorphisms, or genetic markers and things that haven't been reported before, that can really influence the way that treatments come about and are.
Yeah.
And
UK Biobank is probably 88% white people because it's based in the UK.
Yeah.
Like, I mean, to be fair,
fair enough.
there are other databases that are like based in Asia, based in Africa, that are grabbing more of distinct populations there. But the more data, the better. However, I'm going to skip actually to, I think, one of my near to the last stories, because everything that you just said sounds amazing, sounds positive, and sounds great.
What did I do?
Because we're living in a Western democracies.
Exactly. Yeah.
That are using this genomic information for the health industry. The question is it okay to publish or provide this information to an authoritarian government? And more to the point, is it okay to publish Chinese research? So last week, the journal Molecular Genetics and Genomic Medicine retracted 18 papers from Chinese institutions because of ethical concerns. This is thanks to a bioinformatician. I don't know.
Not magician. I mean, they're not statistics magicians. Yeah, informatician.
Infomagician. I'm going to stick with infomagician.
Okay, sounds good.
Eve Morrow, who has been pointing out studies that fail to get proper, free, and informed consent when collecting genetic samples, is sort of being credited with getting these 18 papers taken down. He's especially been focused on vulnerable populations in China. He raised questions about the now retracted papers back in 2021 and is saying that this appears to be the largest set of retractions ever over human rights issues. So the retraction notifications are kind of basic, citing inconsistencies between the consent documentation and the research reported. The authors from China are kind of saying, hey, you shouldn't take it down because it's fine. But there's also, of course, different standard of consent taking place, apparently. The research that was taken down largely focuses on genetic markers that can be used to identify individuals and distinguish ethnic groups. Most of the papers focused on minority groups such as the Uyghur, Tibetan, and Hui people that are subject to intense surveillance by the Chinese security apparatus and may have been pressured into cooperating with researchers. The lack of rigorous informed consent, though, also applies to the Han Chinese majority. This came up, I don't know if you recall, I was talking about a study. It was that didn't have anything to do with genetic markers or anything like this.
No, but we were talking about, paper mills. We were talking about the pay to play.
Yeah, but
research.
This is earlier. This was a study about elder care homes in China, and they wanted to see how they could affect health by reducing sodium by using a sodium substitute.
Oh, I don't remember
that one. And so this study was conducted on these elder home residents. And in the study, the researchers suggested that some of their results may have been compromised by subjects who had detected the lack of sodium in their food additives in their food and began adding salt themselves. And the problem with that is that's not the kind of thing that's likely to occur if you have informed consent and people know that they're part of a research study. So right there in the paper, you had the researchers complaining that the subjects had figured out something was wrong with their food, right? And if you looked through, and I tried to go back and remember and look through the consent documents, and supposedly there were people were signing stuff, but what they cited was they had gotten approval from the municipality, from the region governing. Yeah, they were like, oh no, the government said it was okay. This is what they kind of came back. And so this is sort of perhaps a problem with conducting or publishing science from authoritarian governments, is that if it involves humans, we don't have the same ethical standards taking place that we assume are the guardrails that we assume are in place in Western research.
I think that there's an interesting question there as well, not just with respect to authoritarian governments where there may be separate ethical standards, but are also corporations that have ownership of data. We know in recent history as well, Facebook has used user data to carry out studies about how, and publish studies related to how people respond to certain stimuli, advertisements, whatever, without anybody saying that it was okay for Facebook to use their data, or, it's not anything that particularly could have physically harmed people, but in some cases, these were, advertisements, bits of propaganda, other things that may have influenced mental health or other aspects of emotional health. So I think as we move forward, there is a big question about global research, and there is, I think it's the scientific community's responsibility to have a conversation about that, and for scientific journals to ensure that there is a standard, a minimum standard of consent and ethics. And the United States generally used to have really bad standards. We didn't have consent,
Yeah.
right?
No. You could surprise somebody by studying them.
Right.
Oh, guess what? You're part of a study. Those shocks that you received, we actually did them on purpose.
Yeah.
You
So,
were
and
actually on a very powerful psychedelic. You haven't gone crazy.
you're a
We'll tell you after
spider.
the fact. We needed to
That
study
was
you
weird.
without you knowing.
Yeah.
Just
So,
finishing up this, more
yeah,
notes
but we've come up with new standards that universities have to follow, institutions have to follow. There are, committees that have to okay research. It's not perfect, but yeah, go on.
So the other thing that they sort of noted in this is that these studies have the research teams themselves have sort of a unusually high proportion of investigators affiliated with police and justice systems, which when you say unusually high, that's like almost unheard of in the West to have the police department investigation department actually being part of research studies.
Yeah.
And the fact that a lot of these studies are focused on distinguishing ethnic groups, like, what do you mean distinguishing? Like when we were talking about it, we're talking about adding more diversity to the profile so that we can study perhaps differentiation of disease in different genetic groups. What this seems to be based on is how do you identify, because you have an authoritarian government that is also a ethnic majority in China. And so if they are concerned about something, it looks like the precursor to a genetic-based ethnic differentiation within their society. And it is...
But they don't need to be publishing that. I mean, they could just be like, why? It's like, hey, world, look at this hand in our science research. Meanwhile, I mean, that's the question is, any knowledge gained by the scientific method can be used to all sorts of purposes, right? We hope that science will be, the process will occur in the fairest way possible for the betterment of humanity. But yeah, we don't know. And so how can we help move that forward?
Yeah.
It's a big question.
Yeah.
It's interesting.
One of the things we can do is
question.
stop publishing authoritarian research on humans. What if there's a human involved? We've already complained too much about Chinese research and the quality of what's coming out of there. It
doesn't matter. I mean, just the quality, right? Quality and the credibility. And
the credibility is so heavily questionable for the research that I've been coming across that's coming out of China to begin with. But
there are other places as well. So it's not
for publishing companies who are publishing science in the Western world that has an ethical guardrail for human trials. You have to understand that any paper that you're accepting from an authoritarian government like China does not have those in place. And should you be publishing it? It's a question that I don't think that they have been looking at. Until
philosophically, ethically, it's probably a very important question for the journals to be considering, and moving forward also for them to be considering in also the political aspect of, how and if they're being used as tools for certain political ends, Gary L. says science is self-correcting, and it is over time, but sometimes that takes a lot of time.
Yeah, I mean, you look at what happened with the Alzheimer's hidden mystery protein thing, right? That might have put research off by 20 years because somebody forged data.
but self-correcting.
Self-correcting, but 20 years later, like we
Self-correcting.
want our science and we want it now, and we don't want people messing around with it in between.
Science is for everyone. Science,
Even authoritarian governments. Come
it
on, people.
is. I'm
It's
gonna
for
say,
everyone.
come on, it's for everyone. But but these are good questions to surface. You want to talk about bulls and balls? I got a couple stories about bulls and balls. First story is researchers. Let's see if I can get my stories up here queued up for me. Researchers looking at fertility in bulls. We're having fun trying to figure out how they could determine why certain bulls are more fertile or less fertile than others. And in the reproduction of bulls and these bull markets in the EU, these researchers have basically the markets. They take semen from all the bulls like once or twice a week, and then they water it down a little bit, and it gets mixed up with others, and they give it to females. And sometimes turns out that some of the bulls aren't really fertile, but they have created a system where it's just kind of usually they just expect them all to be fertile, and when they're not fertile, that costs the ranchers money, the people who are trying to work in the sales of these bulls.
Why?
Yeah,
Do they mix?
I don't know, for the betterment of the bulls, of the beef. I
No,
don't know.
because here's the thing.
No, but okay, but they do. Okay,
Like
and
in
so,
just
but what
basic
happened?
animal husbandry, you want to be able to control like the genealogy so you don't get too closely related. So how would you be able to control that if you don't even know what was in the secret sauce?
What was in the sauce? Well, the researchers have discovered just this problem overall is that the bulls, some of the infertile ones slip through because of the way that the reproductive process is managed, and they don't show up during conventional ejaculate screening because the genes aren't necessarily in the sperm or the ejaculate themselves, but actually further back in the testes, in the cells that create the gametes. And so the researchers who just published their story in Nature Communications, they used the testicles epididymis and vas deferens from 118 bulls of reproductive age. They were not killed for the research, but these parts were used for the research. They biopsied them, got the messenger RNA, the transcriptomes that were there, and then looked to see what genes were active in which tissues. They found a whole bunch of genes that were active, and a lot of variants that are related to fertility are also conserved in human fertility. So this study itself is not necessarily trying to help make human male fertility better, but they want to help the bull ranchers save money and have fewer infertile bulls if they can help through genetic screening methods that are a little bit better. But because they have found a whole bunch of these bulls, and it's a lot easier to get these parts from bulls to do this research than it is to get them from humans. Voluntary donors, they don't pop up all the time. It's hard to compare. So what they've come up with, though, is a bunch of genes that are highly conserved in human males that may be helpful to help infertility in human males, and also save bull ranchers money.
So this is the I guess the point of this story too, is that instead of focusing on being bull ranchers, they should have been managing other parts more closely.
Yes, more closely. And so this brings me to the next story of really, really balls. It's hard to study the testes. We have organoids for all sorts of different tissues. We have testes for the liver. We have not testes,
What?
organoids. We have
My
organoids.
God, this has been anatomy with Kiki.
I
Hang
know.
on a second.
I did great at anatomy. Okay, anyway.
No wonder it hurts so much when I get kicked in the liver.
Yes. Anyway, we have organoids for the liver, for the kidneys, for the brain, all sorts of organoids. We don't have any for the testes. And so these researchers um, out of the Bar Ilan University have published their paper in the International Journal of Biological Sciences towards the testes in a dish. Generation of mouse testicular organoids that recapitulate testes structure and expression profiles. So in this work, they used rat neonatal testes first to see if they could take neonate testes and get them to grow in a dish. And lo and behold, they did. And they said, okay, now we don't want to be taking neonatal testes because a lot of the developmental issues that would be influencing development of these reproductive organs would happen earlier. And so they took embryonic tissue to create organoids in a dish. And they were able to pretty much get organoids that grew within a dish structure. They could not get adult tissue to grow into an organoid in a dish. And the embryonic tissue, they were completely unable to get to produce sperm. So these tissues, these organoids, are kind of working. They've been able to get the rat embryonic tissue to create organoids. They last for about 9 weeks, but they don't create sperm, but they start kind of the process. So there's still some stuff that they haven't quite figured out on how to maintain and sustain these testes in a dish and keep them working.
We wouldn't even need the bulls.
Right. Could be used. If we could make testes in a dish, so many things would change in the world. Would they? I don't know. But this system would be a great system, and that's what they're publishing for, is the model system to be able to create a laboratory process to enable the research into this development and diseases and dysfunction related to the testicular reproductive tissue. So bulls and balls. Dun, dun, dun. Researchers are having fun.
If then I could reproduce my testicles in a lab dish, could they then survive in a what was it, like head in a jar situation?
Right.
So, the testicles could live on into the future. Or
your testicles, if you donated. No, actually, I mean, they'd have to, at this point, like I said, the adult testicular tissue that they tried to use to make organoids did not create organoids. They couldn't get it to work. Maybe at some point they will be able to do something like that, but at this point they can't. So it's embryonic tissue only at this point. So that means that we're not at this stage going to be looking at human testy organoids.
Yeah.
It's going to be rat or other animal organoids that where that's more acceptable until they can figure out how to use human adult tissue to be able to make testes in a dish. It sounds like, I don't know, what restaurant in New York City are you going to?
Is that what you thought of? Yeah, my first thought was it would make
Yeah.
it might make divorce court a little bit more friendly. Like, well, we've negotiated. We've decided that yes, Miss Jackson, you can have both of his testicles on a dish, but we're going to do it through science, not through litigation.
Not through castration at all. No. Oh boy. Oh boy. Oh boy. Oh boy. Justin, do you want to talk about Denmark or Africa?
Is it both? Yes. Oh no, it's Denmark. I think is the next one.
Yes, and.
Yes, and a Danish bog body found in 1915 has begun to tell its life story. The remains are a few bones, fragmented skull, piece of wood, have been resting in the rural bog near the town of Vitrup for nearly 5,000 years before it was discovered. Now, research led by the University of Copenhagen has made a thorough assessment of the bones. The man, who is estimated to have been in his 30s when he died, using carbon and nitrogen isotope levels in bones and layers of teeth, researchers could reveal aspects of diet at different ages. So they found that he began as a fisher hunter-gatherer in northern Scandinavian coastal regions, likely either along the Norwegian coast, possibly the Swedish coast, possibly as high up the Norwegian coast as being an Arctic fisher. Then came a change in diet, one that was more dominated by resources from land and freshwater environments. Before the age of 18, 19 years old, he ended up among regular farmers, possibly within a culture that was common in northwestern Denmark and Jutland, where his life eventually ended violently.
Probably because of the farmers that came around were like, we don't need you scavengers anymore. No more.
Well, it sounds like he lived there for maybe a couple of decades, a decade and a half amongst the farmers. But either he died violently either by ritual sacrifice, because archaeologists just can't resist inventing ritualistic causes for human behavior. Although there may also be other evidence of this being more common in those days. Or it could have just been a mundane murder, like you said, like, I don't like that farmer guy coming around. Foreigners. Either way, the skull fragments suggest at least 8 blows to the head, possibly derived by the wooden club, also found with the remains. One thing that stands out from the current findings is the origin of the Vitrup man as being different from the known Danes in the region. Everything from thickness of the skull to the isotopes, the things don't match the locals, but do have some ancient human remain correlations in Norway, possibly Sweden. Either way, though, this is this person traveled to Denmark. To have traveled across 100 or so kilometers of open sea was actually a pretty small task for Vikings and their longboats. But Vitrup man made the crossing as a youth around 3,000 years before even the earliest known signs of any boating in Scandinavia.
Wow.
This is 4,000 years before any truly seaworthy longboats were invented. So, or known to invent. And the oldest known boats are only known because they were not taken by the sea. The wood, right? So there
Right.
are oars and I guess some parts of a boat frame that have been discovered in another bog in Norway that are the, I guess 2,000-ish. Would they be 2,000-ish years old? Yeah, they must be. Somewhere in that range. And it's about 4,000 years or or 1,000 years ago, there was a old boat that is found buried, like as a burial, right? 900 AD ish boat construction that was buried in a mound. So these are the oldest boat remains that we have from Scandinavia. This fella is 4,000 years before all of that, and has traveled this great distance by sea, reinforcing that there is a gap in our historic maritime knowledge that might be kind of serious. An unwritten history of invention and migration by wooden craft that would be disintegrating beneath the waves of water and time, thousands of years before any sort of physical trace could be found.
And there are likely many more individuals like this around the world, right?
And there's
ahead of schedule, right? Ahead of the evidence.
Yeah, like there's even whatever, if it's Homo erectus, or maybe it's a Denisovan, but the areas in Oceania that they've always looked at and gone, there's no way somebody could swim that.
Right.
There's ancient hominins on islands in the Aegean Sea even. Doesn't make sense how they could have gotten there. Well, it does if people have had boats for a really long time and we just assumed they didn't because we couldn't find any of the remains. This is a sort of interesting way of determining that. And you find a human who's like really far away from anywhere they should be.
And
but
was beaten
really
to
close
death.
if you went by speed. And beaten to death. Well, that helps too. If you're going to do a murder and we want to get away with it, don't put them in the bog.
Not the bog.
We're going to find the evidence.
Bogs are good at preservation, everybody.
Dun dun.
Dun, dun.
We've concluded the investigation into the bogman.
Tell me about ADHD then.
Oh, you want to go third story? Okay, here we go.
Oh, do you want to do that right now, or did you want to save it for later?
We can save it for later. We can do it either way. I'm happy either way. You want to do it now? You want to do it now? You want to do it now?
Whatever.
I got it right now.
It's on the list.
Okay. This is a neuroscientist and psychologist from the Perlman School of Medicine and the Indian Institute of Technology, suggesting that early hunter-gatherer groups may have benefited from the behavior of individuals with ADHD. They think these individuals were more likely to explore beyond known territories, leading to the discovery of new game to hunt or plants to eat. And it says, according to this, people with attention deficit hyperactive disorder tend to have more trouble focusing on activities around them. They may also exhibit restlessness and be easily distracted.
What?
Prior research suggests that the condition is inherited and it may have been affecting humans for many thousands of years. In the study, researchers looked for genetic advantages of ADHD, and they had experiments involving 457 volunteers playing a berry picking video game of some sort, hovering a cursor over a berry bush for several seconds and moving the cursor as clicking on stuff, I guess, to harvest the berries, and then you got to wait for them to regenerate. But then you can also go around to other berries, and then you got to hover the cursor over there for the berries to pick. It's one of those things that sort of, it sounds like a sunk cost. What is it? What is that called? Sunk cost?
Yeah, the sunk cost fallacy, or it's like when you choose a lane in traffic, and then you're like, that lane over there is going faster, I'm going to move over.
Yeah, or if you go, well, I took all the trouble of getting into this lane, I'll just let it sort itself out.
Or I'm at the grocery store and I've been in this lane so long, I'll just stay here. Just gonna keep it.
Yeah.
Do it.
So apparently they've discovered that the folks with the ADHD were much more likely to ignore sunk cost fallacy and much more likely to do the lane change or the line change and switch to other bushes in this game to pick berries from.
That would be me.
And their strategy was successful. They tended to collect more this way than the groups who were being more conservative about it.
I'm not surprised. I mean, there are okay. Evolutionary psychology. Every time you get into these kinds of things, it's like, and we're making up a story based on what we see here in the now, and blah 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 blah. But the truth of it is, a lot of the things, not all of the things, a lot of the things that are present in our current day form, function, behavior are there because they were useful in the past. So how would ADHD be useful? Maybe, people really great at like 80% finishing things, and then you're like, yeah, okay, time to move to the next thing, and you leave before you use up all the resources, and get people to try something new. Maybe it was helpful in that way. And then you have the sociopaths and the psychopaths who are really good at like going and killing the other tribes,
Yeah. We call them, in the olden times, we called them kings.
Yes.
Tribal
now
leaders.
we call them CEOs.
Now they call CEOs and world leaders. Yes.
Yeah. I don't know. I mean, it's an interesting... the test is, hey, do you have ADHD? And then, then they made up a story about why the restlessness would be beneficial in the past.
Well,
but
it's an interesting thing because just aside from them trying to connect it to early man and trying to like figure out what advantages there could be in resource gathering, which is what they were trying to look at. They were just trying to look at how would this affect a simplified version of resource gathering. But what I thought was really fascinating is, to me, that makes ADHD make so much more sense if people with ADHD lack that the feeling of consequence of a sunk cost.
No, it's novelty. It's dopamine. You got to move on to the next thing. You got novelty.
Maybe that's what's being told all the time.
It's not great for the capitalistic culture that we are existing in currently. But it's also great for, being the kind of a creative, being an entrepreneur, maybe being, somebody who is able to try new things, come up with great new ideas. But yeah, there's multiple sides to it and multiple different neurodivergences, and so if there's no one type of ADHD, there's a huge spectrum. So.
But that's all said. If there is a lack of sunk cost, appreciation of that sunk cost, or feeling of that sunk cost fallacy, that I've already invested time in this idea or this project, and that you can simply walk away from it without feeling like you've invested, you're invested in this line, and now you've already committed to it. You're like, no, actually, I could just walk anytime. I can move anytime. Then it sort of makes sense how people can go unfinished project onto something else, unfinished thought, talk about something else. The lane changing aspect. If you don't have that, or just idea, and when you talk about like in creatives and that sort of thing, here's the way that we all do it, and here's what you've been trained in school. Yeah, that's great. I got this other idea. I'm going to go try that. Like, a lot of it could just be not feeling like you owe the line anything, that you owe that the
no, because something else is more interesting over there, or that one looks better, or like that. I like that one better now. I'll go there now. But then human psychology is more complex than that, so you can't just say, But yeah, maybe for some people, that there is no sunk cost. Maybe there is no feeling of regret or concern over the switch.
Yeah.
Yeah.
That makes a tremendous amount of sense.
People are different. They're very different than AI.
Are they? I thought they were getting more and more similar.
Yeah. Well, yeah. Yeah, that's kind of what this study is coming. Researchers, surprise, surprise, published in PNAS Nexus. Their article, How Persuasive is AI Generated Propaganda? It's persuasive. In their study, they used articles that had been identified as propagandist online in the past. They used ChatGPT-3, trained it with the articles, and they used very specific researcher-written thesis statements that they fed to ChatGPT for each of the for shorthand for the articles. And in doing that, then they got a bunch of people to read articles that were then rewritten or created by ChatGPT related to these topics and what had been fed to them by the researchers. And they determined that the propaganda from the ChatGPT was pretty much as good as, or close to as good as, the original propaganda. And then when they got people involved to kind of edit the propaganda, then it got even better. So when people with editing, propaganda by AI is even more convincing. so it's pretty amazing, everybody. AI
maybe more than one
is good at propaganda based on the propaganda that people have been giving themselves for years, and when humans try to make it better, it gets better. Who knew?
There are only two forms of intelligence. There might be like 50 kinds of ADHD. Fine, whatever. There's only two kinds of intelligence. Intelligence that is binary and intelligence that is not. And it looks like when they work together, they can make good propaganda.
Hey-o. All right. Anyway, yeah, who knew AI could possibly be used by people for particular propagandist aims? This is This Week in Science. Thank you for joining us for this week's show. It's so exciting to have Justin back on the show today. We are going to finish up fairly soon here, but before we do, I have our last stories. I just want to say thanks. And if you are enjoying the show, please share This Week in Science with a friend, and head over to TWIS.org if you are interested in seeing our show notes and other stuff related to past episodes. We also have ways for you to support the show. So if you click on the Zazzle link, you can buy merchandise that is TWIS related, and the royalties go back to help keep the show going. There's a lot of art related they're products that Blair made. And then there's a calendar. If you know, it's February. Oh my goodness. But there's a calendar that Blair made if you want to get a calendar from Zazzle for This Week in Science. And also our Patreon is at twist.org. You can click on that Patreon link and become a supporter in an ongoing fashion, monthly. Choose your amount, $10 and more a month, and we will thank you by name at the end of the show. We can't do it without you, so thank you very much for your support. We're going to come back right now and talk about a few more little science items. I'm going to jump into a story about brains, because you know how much I love the brains. So let's talk about how none of us would be here running around, talking, discussing, doing anything that we do if it weren't for a virus. Realio, trulio, we are the product of a virus that infected our lineage at a time way back when. What's called a retrovirus, endogenous retrovirus. There are a number of them. They're called the ERVs, and the ERV1s are involved in a lot of aspects of our genetics. This particular study that was just published in Cell It's called a retroviral link to vertebrate myelination through retrotransposon RNA-mediated control of myelin gene expression. Blah, 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 blah. What does that mean? A long, long time ago, a virus that goes backward, it goes into the DNA to get itself locked into the DNA. So then our cell DNA transcribes it so that then it can take over cells and do things. It tried to do that, but then it got involved in the process of making our nerves work, which is, I think, one of the most exciting aspects of this. Myelin is responsible for the ability of our nerves to send rapid signals down their lengths, down the axons, from our heads all the way to our feet. And in this, myelin is really essential to the fast action that we have, the fast responses that we have. And this study, they discovered this endogenous retrovirus that they have named retromyelin. Very, very flashy name. Retromyelin is found in a lot of different species, a lot of different groups of vertebrates. They've found them in fish, in amphibians. They have found them in jawed vertebrate species. They found them all over the place. And so they're like, okay, all right, retrotransposons. These are really interesting. They're known to be a huge part of our genome. But for this aspect of our genome to be so important to such an aspect of our ability to function is one of the most exciting, interesting aspects. Second, they did a sequencing and phylogeny comparison. So they started looking at retromyelin in a number of different species. They determined that it wasn't one infection that was just carried through all of us. This is convergent evolution. This is a number of different infections. So one of our common ancestors was infected, and then maybe we got infected again later. This retrotransposon, retromyelin, whatever it is for us as people, is very slightly different than the one in birds. It's very slightly different from the one in reptiles. They're not the same. Yeah, so it's evolved separately. It is this virus that allows a cellular process to take place that creates an oligodendrocyte that wraps around our nerve cells and in a certain way to allow efficient nerve impulse, action potential transmission to take place. It's happened multiple different times in different species. It's not just once. And so we have like these really, really tight wrappings that make our axons, our myelin, really efficient versus other species that don't. So it's different.
But like how is that possible?
I don't know. This is so exciting to me. Yep. So jawed vertebrates, jawless vertebrates, invertebrate species, we've got all over the place. And there's this incorporation has occurred over and over and over again. And when they tried to take it out, through genetic modification, the oligodendrocytes just didn't function. They didn't make myelin. It didn't happen the way it was supposed to. So it's this retromyelin, this endogenous retrovirus, this very specific derived element is essential. So, but it's essential across the board and differently.
Okay, so here's the question then, because first of all, that's not possible.
But what else? Okay, so
hang on. hang on.
How do you know?
Deep breath. Deep breath. It's not possible because the way evolution works, that it wouldn't have shown up in a later species or a different branch of a species and not had been there before. However, what we then might be looking at is, it could be replacement. Like you get there's a right, we found a better wrapping virus and it comes in and takes over the job. Is that what we must be talking about?
Right. So this is a question that the researchers ask themselves, and they say, "Hey, we know this is wild, right? That this co-option that it occurred over and over again in this convergent evolution way. Each host was separate and adapted separately, uh, and that." Yeah, they explored the different species, but they do say, okay, we don't know. There are limitations to what we've looked at. All we've really looked at are molecular relationships. There's specific requirements, and we've just found jawed vertebrates. I mean, I'm looking through the discussion right now, but
one of the other things that then pops in my mind is like, okay, if it didn't actually happen multiple times, the alternative is that a virus was incorporated very early, and the viral DNA is just our DNA.
Right.
Right? We're not looking at something separate and outside that's joining the game. It's like, no, some of the essential building blocks of life, intelligent life on planet Earth, or cellular functioning life on Earth, life on Earth is it's virus based.
Yeah. So they say, there is possible, multiple mapping. There might be ambiguity. There's possible challenges with specifically determining specific genomic location within long sequence reads. There's, what loci is it found in? Multiple mapping is possibly there. There's evolutionary time, so like younger sequences versus older sequences, and how do you pinpoint that kind of stuff. So there's more work that needs to be done for sure, and they acknowledge that. But they're like, hey, this is what we found, and this is what it looks like. So it's like, this is a challenge now, I think, for for researchers to either confirm or
because it's obviously not how it could have happened. Like, there's just not
over and over and over again.
Well,
Why?
I mean,
how?
like the thing is, it would be like cellular function was not taking place in several species until no, that's not one of the possibilities. That's not one of the options. That can't be.
Yeah, and why is it a very specific interplay between this retromyelin gene and its all of its variants, and very specific other molecules within pathways within our nervous system, and also other vertebrate species nervous systems. Why does it work in the way that it does? So yeah, I don't know. We'll see where it goes. Why was it co-opted? How did this happen? Is this real? Is this even real?
Yeah. Is this even real? Well, I mean, in the genome that we think of as the blueprint, we've also since discovered it's not so much a blueprint, it's not so much instructions, it's just raw ingredients. It's a list of ingredients. It's a list of the building materials that the life form can use. And so if that building material was sourced at some point from a virus, makes no difference. It's a building material now, and we're going to use it however we need to.
For sure. But if that it's like, though, with our computers, we need very specific, USB-C now versus just regular USB or Lightning or, whatever the standards or the differences, whatever is being used at a particular time. And it's kind of like this retromyelin fit along the way, right? As the standard changed, the species, the infection changed, and it was still able to fit in the right place to influence myelin. And this is the
Yeah.
question of how did that happen? Yeah, the likelihood. I don't know.
Yeah.
I want more people to study it. That's why I think it's so fascinating. Let's talk about it, everybody. My last study, though, is about music in the brain, and we've talked about music and appreciating music before. And this group out of UC San Francisco has studied the way that the brain responds to speech and phonemes, and found that there are very specific neural groups, neurons also, that respond specifically to certain phonemes, which are and pitch changes. It's certain sounds in speech.
Oh.
And this time around, the researchers were like, okay, so let's turn it not just from speech to music. So not just understanding speech and appreciating speech, but music itself. And they found that just like with speech, there are specific neurons that are listening to the pitch, to the melody, and making predictions. And there are some of these neurons that are very similar or the same as what are used in listening to speech. But there's a specific population of neurons that is predicting how the melody is going to change or the pitch is going to change. And that is a very, very specific set of neurons in the brain that do that. And so this may be not just part of how we appreciate music, or how we, when you wait for that kind of like completion, if something goes from a major chord to the minor chord, and then you want it to come back to the major chord, or to finish with in a nice way. You want completion. We've learned certain things.
Some of that, of course, is going to be training,
Yes.
right? Because depending on what kind of music you've heard, you have a different expectation of what should be next, and then that becomes your taste in music, or the flavor of music that you sound of music that you like.
The Sound of Music. That was a movie with Julie Andrews. Anyway, so it's pretty interesting. These the researchers being able to look at the brain and using people who were undergoing surgery for epilepsy, had them listen to Western music, modern Western music, bunch of different things, and then recorded with electrodes on the surface of the human auditory cortex. And we're able to see how the melody was encoded as the music was played. And they were able to see that different neurons were more or less excited or stimulated by the changes in note, by the changes in pitch, and those were all involved in the way that the hearing and the appreciation occurred. They moved beyond just talking in sentences. They looked at music. I think this is very fascinating because there is, I think there's something to be said for the way that people speak and the tone and the pitch. Like if you listen to Martin Luther King Jr.'s I Have a Dream speech, the way he starts, and the way he starts with a slow and kind of low but major chord of his voice. And then it starts to increase, and the pitch changes and the tempo changes, and he brings you along almost in a musical way until he comes to the completion. And it's like the great speeches and the great orators in our human history use the musicality of our voices. And I think, I think there's a definite relation between, our understanding of speech and our appreciation of music.
Also, the whole repeated phrase thing.
Yes.
Like if the brain
repeated.
if the brain has an expectation that they're going to hear it again and then it's fulfilled, it also tends to ring truer if the brain is predicting what's going to be said and then it's said. You're like, I knew it. That sounds right.
Yeah. So anyway, our brain isn't just looking for notes. It's actually like going, hey, I think I know what's going to come next. Is it going to happen? Is the melody going to happen this way?
Kind of interesting if they, I would like to see what happens if they, when it was like, they were given like a classic country song, like what goes up must come down, what goes around, tuna fish. And you see, like if the brain's like, what happens when the prediction can't come true? When the prediction doesn't fit, there's the brain like, is like looking back at, wait, you said down over here, and now you're came back to tuna fish. That doesn't sound right at all.
What are you doing?
What happens to the predictive ability of the brain then?
Yeah, there are lots of studies like that, though, where they, but usually they're visual auditory connections where they have, it's a red square and somebody says the word green. And, and so it asks you to try and figure out the difference between. So there's a bunch of work that's been done in those kinds of, how the brain differentiates between these sensory inputs. But, in this case, as we well know, our brain is a predictive instrument and it's doing abstract processing with very specific neurons that are predicting music and melody and meaning for emotion. It's great. Do you have one more story or do you have to go?
Oh no. This is a story about how I got absolutely and completely gazeboed the other night. Oh my goodness.
Why do you keep saying gazeboed? That's not even a thing.
No, but you could probably tell what I was talking about.
It's questionable. I don't know.
Okay. What if I said plastered, blasted, loaded, schnunkered?
Those are kind of Western Americanisms, yeah?
How about schnooked?
Schnookered. Isn't that a pool phrase? No, I don't know.
Pissed, hammered, wasted? Basically, what happened was some German linguistic professors looked at the way that Americans talk about getting drunk. And they figured out that basically, by the time a youth in America is coming to adulthood, they've heard references to getting drunk ending in an ed so many times that if somebody says, I got totally pajamaed the other night,
it doesn't matter what you say. It's just
as long as it ends in ed, it means drunk.
Oh no!
Drunkenness is so prolific in the United States.
It's not just the United States.
No.
But I'm glad this is that now what they can, how the Germans can see us, the drunkards.
But can you like think of all the different ways?
I get so Americaned.
Because what was it? Was it that partial list right there? It was just like wasted, blasted, hammered,
car parked,
pissed. Car parked? Is that one?
Yeah, car parked.
I've never heard of it. That was car parked. I totally car parked last night. Oh my goodness. Fudded? Oh, I got totally fudded. Right? Like all of this, it's like universally, like it ended in ED, and therefore it meant drunkenness. Anyway.
And I really love this though. But the word though is that it's not a synonym. It's a drunkenym.
Drunkonyms.
How many drunkonyms can you name in the next 30 seconds? The loser has to take a shot. Ugh.
Anyway, that was all that story had.
Well, thanks for the
Splunkered.
drunkonyms.
Dog tickled.
Schnookered, right?
Okay,
Totally
I like that.
schnookered.
Schnookered, I think means you got fooled.
Right.
Wait,
Isn't
maybe.
that's what I thought. Isn't that like a pool term or something?
Yeah.
Ah, Schnago. I'm gonna get so scienced. That would be fermented. Anyway.
Maybe that's where it all started. Fermented.
I'm looking at the abstract right now, and I'm going to share it on screen for our video viewers because they share a quote at the bottom of their abstract. Obviously, Americans, like many other peoples of the world, have taken being drunk very seriously. Harry Jean Levine, 1981. Yeah, thanks. Okay.
So America was one thing that almost lost the Revolutionary War, the fight against the British. George Washington's troops almost abandoned serving because there was a daily ration of gin that they couldn't provide anymore. They ran out. They switched it to whiskey. And the troops were like, okay, but we need a little bit more because that's not what we were expecting. But they had a daily booze ration. And that booze ration, if you look at it, was like, I've met a few alcoholics in my day.
Hey.
And even they would have looked at the amount of booze being drunk by the average American soldier in the Revolutionary War and gone, geez, that's a little excessive.
Oh my gosh, some of these are okay. So here are some synonyms. BBC Culture article by Dent in 2017 states there are 3,000 words English currently holds for the state of being drunk, including ramsquaddled, obfuscated, tight as a tick, and the curious been too free with Sir Richard. This is a very fun study. Thanks, linguists from Germany. Yeah, at certain points in time, booze was safer than water. In a way, yeah.
Oh yeah, you know, good point. It's the only way to keep keep the dysentery away from the troops. To drink booze instead of water. We'll worry about marching in a straight line later. We're not going to spend too much time. Oh, maybe that's why they spent so much time actually practicing marching. That's probably why they weren't just practicing like being a uniform group. They were like,
oh no,
all right, when we get out there, you're going to have to be able to walk
together,
straight-ish line with this much booze in your belly. Oh gosh,
splonkered, yes, dogged. I love all these.
In fact, when that famous painting of George Washington crossing the Delaware, to go invade the fort, whatever it was over there in Washington D.C. or wherever that takes place.
Yeah, once upon a time.
Delaware, is that the D.C.? I don't even know. They took the fort from the position from the British because they showed up after some holiday. And the British were all passed out. So that noble look of going across the raft and to do the invasion. They showed up and everybody was unconscious from over drinking.
That's the
everybody
best.
Was, and
that's
they
the
just
best time to attack. Yeah.
Hey, you guys were sleeping, but we just won the big battle. What big battle? Oh, the one.
There's no battle anymore. You all were sleeping. Okay. We took over. Good night. Good night.
Oh,
back
man.
to your quarters.
Right.
We're in
charge
we'll
get you next time. Okay.
I hope we catch everybody here next time.
Yeah.
Yeah. We've made it to the end of another great show. Yeah?
Yeah.
Oh, good. Well, everyone, thank you so much for being here. Thank you for listening. And we really hope that you enjoyed the show. I have shout outs to our chat rooms. Everyone who is here chatting during the show, thank you for your comments and your statements and your questions and your little side battles taking place in various places, YouTube, Facebook, Twitch, Discord, all these places. I see you. Fada, thank you so much for your help with show notes and social media, getting all the word out about This Week in Science and the stories that we cover. Gord, Aaron Lore, other people involved, thank you for helping to make sure our chat rooms are pleasant places to hang out for all the peoples. Identity 4, thank you for recording the show. And Rachel, thank you for editing the show. And as always, I definitely could not go on without thanking our Patreon sponsors. So thank you to Aaron Anathema, Arthur Kepler, Craig Potts, Mary Gertz, Teresa Smith, Richard Badge, Bob Cole, Kent Northcote, George Corris, Pierre Velazar, John Ratnaswamy, Carl Kornfeld, Chris Wozniak, Vegard Chefstad, Donathan Styles, a.k.a. Don Stylo, Ali Coffin, Regan Shubrew, Sarah Forfar, Don Mundus, B.I.G., Stephen Alberon, Daryl Myshak, Andrew Swanson, Fredus 104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles Jack, Brian Carrington, David E. Youngblood, Sean Clarence Lamb, John McKee, Greg Riley, Mark Hestenflow, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Richard Brendan Minish, Johnny Gridley, Remy Day, G. Burton Lattimore, Flying Out, Christopher Dreyer, Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul Rick Ramus, Philip Shane, Kurt Larson, Craig Landon, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, Lon Make, CEO, Adam Mishkan, Kevin Parachan, Aaron Luthen, Bob Calder, Marjorie, Paul D. Disney, David Simmerly, Patrick Pecoraro, and Tony Steele. Thank you for all of your support on Patreon. And for anyone out there who would like to join us as a Patreon sponsor, please head over to TWIS.org and click on the Patreon link. On next week's show,
we will be back Wednesday, 8 p.m. Pacific time, 5 a.m. Thursday, Central European time, something like this.
Wherever you are, whatever time.
It's two shows, but somehow we get them done at the same time. I don't
No, it's
really
one show, same
tectonics.
time, different time zones. Oh my goodness. Want to listen to us as a podcast? I hope you do. Search for This Week in Science wherever podcasts are found. And if you enjoyed the show, please get your friends to subscribe too. Really, take their mobile devices or whatever with their podcast apps and be like this and subscribe them. That would be awesome. But I'm not telling you what to do because no, I'm not going to do that.
For more information on anything you've heard here today, show notes, links to stories, and instructions on how to add us to your friend's device are available on our website, www.twis.org, where you can also sign up to get a newsletter. But it won't be from us. From us?
It's from us. Of course it is.
From us. Oh.
Yeah. And you can also contact us directly through the emails. Yes, I know. The old communication technology. Email me at Kirsten@thisweekinscience.com. Justin is twistminion@gmail.com. Blair's blairbaz@twist.org. And if you do email us, make sure you put twist somewhere in the subject line so that your email doesn't get spam filtered into, I don't know, propagandist AI or I don't know, Saturn, where it's just stuck on the surface of Titan and nobody's really finding it anymore. So yeah, don't do that. Twist, subject line. Yeah.
You can also hit us up on other social media where you can find at Twist Science, at Dr. Kiki, at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
Yeah. Hashtag Sonnets for Science. And we will be back here next week, as you know, and we hope that you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head with a virus.
This Week in Science. This Week in Science. This Week in Science.
This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.